coast stretches quiet under a low sky. It is the world of 10,000 years ago. The last ice sheets have withdrawn northward, sea levels are still rising, and the shoreline of what will someday be called Southern Europe does not yet lie where it will. The Black Sea, sealed for millennia, has only recently been flooded again by the Mediterranean, and its waters are still creeping into ancient river valleys. The air tastes of salt and wet stone. Behind the beach, mixed forest presses close. Oak, pine, the first hazel thickening after the cold, band of Mesolithic hunter. Gatherers, no more than 40 people, has made camp at the tree line. Their shelters are hides stretched over driftwood. Tools are flint barbs and antler harpoons. Their language, not a word of which survives, is fully modern. They tell stories, they keep track of kinship. They watch the stars. A small fire burns at the edge of the settlement. Around it, perhaps 20 figures sit close against the evening damp. Their faces, lit from beneath by the flames, are dark. The deep, rich brown of skin that has never known a winter without full melanin. Their hair is black or near black, their irises, the warm amber dark that every human eye had been for nearly 300,000 years. Firelight catches the wetness in those dark eyes, making them gleam like polished stone. In the centre of the frame sits a child, no older than 4 or 5. Her skin is the same deep brown as everyone around her. Her hair falls dark and unkempt across her forehead. She stares into the fire. Without speaking, a child lost in the private world. Children inhabit before words find them. Camera holds on her face long enough that the viewer begins to notice something. Is not quite what it should be. Her nose, her mouth, the shape of her jaw. All familiar, all echoing the faces of the adults beside her, but something in the way the flames reflect from her eyes pulls the gaze. Her eyes are pale, not brown, not the deep amber black that every other face in the firelight reflects. Her irises are the colour of shallow water in winter, a fragile, clear blue that seems to have arrived from somewhere else entirely. When she blinks, blue vanishes for a fraction of a second behind lids, the same dark brown as her mother's, and then reappears unchanged. She does not know she is different. She has never seen her own face. Nobody has told her that the colour of her gaze is unprecedented, because no one around her can name what they are seeing. In their language, there was and could be no word for blue as a human trait. The concept had not been invented because the thing itself had never before existed. People beside her have noticed, of course. Her mother, when the child was an infant, once held her to the morning sun and studied those pale irises, searching for some sign of illness or omen, and found nothing. The elders glanced and murmured, and in time stopped glancing. She was simply the child with the strange light in her eyes. They did not cast her out. They did not worship her. She was fed, she was held. She learned to walk and to mimic the sounds of her people. The only difference in the whole of human prehistory was that when the firelight struck her eyes, it struck something that had never been struck before in any human face. She is a child sitting by a fire watching the flames, and every single person alive on Earth today who has blue eyes is descended from her, or from someone genetically identical to her, carrying the same accident in the same gene, at the same moment, at the same sat, in the same small corner of a world that had never seen such a thing before. Let the weight of that fact settle, not approximately, not metaphorically. Genetically, The blue eyes in a classroom in Dublin, in a fishing village in Estonia, on a mountainside in Afghanistan. They all trace back through an unbroken chain of mothers and daughters and fathers and sons to one individual in whom a tiny molecular switch threw for the first time. The chain is roughly 400 generations long. In the grand sweep of evolution, 400 generations is nothing. It is a single afternoon. Roughly 600 to 800 million people alive today carry 2 copies of that switch. 8 to 10% of the entire human species. Direct genetic line, 10 millennia deep, running from a single child by a single fire to 500 million faces, spread across every continent. The number defies intuition. Blue eyes feel ancient, a mark of deep European ancestry, trait so old it seems to belong to the landscape itself, to the cold grey seas and the long northern twilights. We have painted them onto portraits of medieval kings and carved them into statues of gods. We have written them into sagas and used them to set one people apart from another. Cultural imagination has made them a symbol of something timeless, as though the pale-eyed warriors of Tacitus's Germania carried a lineage stretching back to the first humans who ever walked the ice frontier. The truth inverts all of that. Blue eyes are not ancient. They are one of the youngest traits in the human repertoire. The mutation that creates them appeared roughly 10,000 years ago, after the last Ice Age had released its grip on the northern hemisphere, after modern humans had already been walking the Earth for 290,000 years. In a species 300 millennia old, blue eyes have existed for the last 3 to 5% of our history, everything we think of as the deep past. The cave painters of Lascaux, the first migrations into Australia, the hunters who crossed the Bering Land Bridge into the Americas. Every face in that long prehistory looked out at the world through dark brown eyes. Blue did not exist anywhere on Earth until this child opened hers. The revelation does not stop with eye colour. If a single mutation, random and neutral, can spread from one person to 800 million in less than 10,000 years, then the entire palette of human physical difference is not a record of ancient, fixed divisions between peoples. It is a live process, fragile and recent. By the end of this story, you will know not only how a tiny genetic switch turned brown eyes blue, you will understand why human appearance is a far younger and a more provisional thing than you have been told, and why the face in the mirror is not a relic of deep time but an echo of accidents that are still unfolding. Consider the world before that child. For nearly 300,000 years, no human being on Earth had blue eyes. The species that emerged in Africa around 300,000 years ago, the species that would eventually name itself Homo sapiens, looked out at the world through irises dense with melanin. Every eye was brown, every single one, the deep, protective brown of the equatorial sun. This is not an inference from a single skeleton. It is the baseline written into the human genome itself. The ancestral state of the genes that govern iris pigmentation is high expression of melanin in every population that stayed close to the latitudes where our species originated. That ancestral state remained intact. There was no variation to speak of, no pale-eyed minority, no recessive frequency hiding in the gene pool, waiting for its moment. For the first 290,000 years of our existence, human eye colour was a fixed trait. As Fixed. Fixed as the number of fingers on a hand, unchanging as the shape of the collarbone or the curve of the heel. Around 70,000 to 50,000 years ago, small groups of anatomically modern humans began to move out of Africa. They crossed the narrow sea gate at the Bab el-Mandeb or walked the Sinai corridor into the Levant. From there they threaded east along the coast of southern Asia, moving in bands of perhaps a few dozen. These were hunter, gatherers carrying their genome, their tool traditions, their language, and their dark brown eyes into landscapes no human had ever seen. Every one of them had high iris melanin. Mutation that would one day produce blue eyes had not yet occurred. If it had not survived long enough to leave a trace. The expansion carried a single eye colour into every latitude. The deep brown of the African cradle, now looking out across the monsoon coasts of India, the open steppes of Central Asia, the dense forests of Southeast Asia, the long walk into Australia. After tens of thousands of years of this slow drift across continents, the species reached Europe. Date is roughly 40,000 years ago. The landscape was a frozen mosaic. Mammoth steppe, reindeer tundra, the scoured valleys between ice sheets. And the people who moved into it were small, mobile groups of hunters. At Peștera Cuoși in Romania, jawbone, dated to about 40,000 years before present, yields DNA that confirms dark eyes. At Kostenki in Russia, skeleton from roughly 37,000 years ago tells the same story. OASE 1 and Kostenki 14 are not hypotheses. They are named individuals. Their genomes have been read, and their iris colour has been genetically reconstructed from the pigments their own DNA encoded. Brown, dark, identical one trait in this one trait to every other human who had ever lived. All known early European Homo sapiens remains that have yielded usable DNA point to the same conclusion. The iris was richly pigmented. The trait had not yet changed. For almost 300 millennia then, the human face wore a single eye colour. Every hunter who followed a reindeer herd across the Mammoth Steppe, every woman who gathered shellfish on the shores of the Red Sea, every child buried with ochre and beads in a cave in what is now Israel, brown eyes. The uniformity is difficult to hold in the mind because it has been shattered so thoroughly. We are surrounded now by a kaleidoscope of irises. Green, grey, hazel, amber, the whole blue spectrum from pale ice to deep slate, and it is easy to presume that this variety is ancient. It is not. For the overwhelming majority of the human story, every face you would ever have seen looked back at you through the same dark iris. Imagine every human face across all of history sharing one NASA eye colour. The painters of Lascaux, standing in flickering lamplight, their hands applying pigment to rock. Their eyes were brown. The hunters who crossed the Bering Land Bridge into the Americas, moving through an unpeopled continent of colossal mammals, squinting against the glare of sun on snow. Brown eyes. First Australians walking the shoreline of a landmass they had no name for. Their skin and hair dark, their eyes the same deep brown as their ancestors who had left Africa tens of thousands of years before. All of them dark-eyed. The uniformity was total, and we have entirely lost it. There was a reason this trait held so steady for so long. Under intense African and equatorial sunlight, a low melanin iris was not a neutral variation. Melanin in the iris absorbs and scatters incoming light. It reduces glare, the painful, vision, washing brightness that makes it hard to track a moving animal, or red the flicker of a distant fire. Without enough melanin, the eye is flooded with unfiltered light, and the world turns white and featureless. More critically, melanin shields the retina from cumulative photic damage. A pale iris transmits more light into the back of the eye, years of exposure under a high UV sun that translates into an elevated risk of retinal injury. Phototoxicity, macular degeneration. These are slow processes, but over evolutionary time they become a filter. Mutation that dialed down iris melanin in the tropics would have left its carrier squinting against the light, vulnerable in ways that mattered for survival. Gene flow from the central populations carried the ancestral, protective version of the iris, genes outward, into every colonized latitude. Even when humans moved into higher latitudes where sunlight was weaker, high melanin baseline persisted because it was not strongly selected against, and because the mutation that reduces OCA2 expression simply had not yet appeared in a viable form. The genetic background of the first 300,000 years was not friendly to the switch that would, one day, produce blue eyes. The environment pushed hard against it. The ancestral iris pigmentation was not an accident of drift. It was a settled solution to a physical problem, and it held for longer than the entire span of recorded civilization by a factor of 50. The story of blue eyes, when it finally begins, is not a story of deep time. It is a story that starts at the very end of our species' history. Then something changed. In one person, in one moment, the mutation survived. The switch that had never caught finally held. Child opened her eyes in a world that had never seen their colour before, and the unbroken brown of ours, species. The uniformity that had stretched from the earliest Homo sapiens in the African Rift to the last hunter-gatherers of the Mesolithic was broken. The world has never looked the same since. A blue eye contains not one grain of blue pigment. None. Zero. The human iris has no blue-producing cells, no blue molecule, no blue structure laid down by any gene. Colour, you see, when you look into a blue eye, is not there. It is a trick of physics performed on tissue that is, in its substance, nearly colourless. A brown eye is brown for a straightforward reason. The anterior layer of the iris, the stroma, a sponge-like scaffold of cells and fibres that sits just behind the transparent cornea, is packed with melanin granules. Eumelanin, to be specific, the same dark pigment that colours skin and hair. In a typical brown iris, melanin covers roughly 93 to 95% of the anterior surface. Light enters the eye, strikes this dense melanin sheet, and is absorbed. The wavelengths that reflect back are what we register as brown, warm and deep, the colour of rich soil. Blue iris has almost none of this anterior melanin. Near zero. The stroma is translucent, a pale mesh of collagen fibres and water. The only dark layer remaining is the posterior pigment epithelium, a thin sheet at the very back of the iris that acts as a light-blocking curtain, preventing stray photons from bouncing around inside the eye. So what happens when the melanin is gone? Light enters the transparent cornea, passes through the anterior chamber, and strikes the stroma. The collagen fibres inside it are spaced roughly 1 micrometer apart, a dimension that happens to sit within the range of visible light's wavelengths. When incoming photons hit this mesh of fibres and water, the shorter wavelengths, those at the blue end of the spectrum, around 400 to 500 nanometers, are scattered outward in every direction. The longer wavelengths, reds and yellows, pass straight through and are absorbed by the dark posterior epithelium. An observer looking at the eye sees only that scattered light, blue. This is Rayleigh scattering, the same physical process that makes the daytime sky blue. Sunlight, striking gas molecules are smaller than the wavelengths of light, scattering the short blues across the whole dome of the sky. The same process, at a smaller scale, that makes shallow water over white sand appear turquoise. The water is not blue. The sand is not blue. Physics of light passing through a medium of the right density and particle size creates the colour in your perception. Blue of a human iris is a structural colour, a physical effect of light interacting with tissue architecture. No animal has ever evolved a blue pigment for the iris. Every blue eye in any species you have seen, a Siberian husky, a Scandinavian child, mountain-dwelling snow leopard, is blue by scattering, not by chemistry. Mutation that makes blue eyes does not touch the OCA2's protein coding sequence itself. It sits in a regulatory region of a neighbouring gene called HERC2, a segment of DNA that acts as an enhancer, a genetic switch that turns OCA2 up or down in specific tissues. The specific change is a single nucleotide polymorphism, RS12913-832, single letter in the 3 billion letter human genome. Swapped from an adenine to a guanine, one base pair change prevents the enhancer from recruiting the transcription factors that would normally bind and ramp up OCA2 expression. The switch is stuck in the low position. OCA2 protein is still made. The gene is not broken. It is simply produced at a fraction of its ancestral level. The melanin factories run slow. The stroma never fills with pigment. Picture a dimmer switch on a wall. The light is still connected, the circuit intact, but the dial has been turned from full brightness down to a faint glow. The brown that once saturated the iris has been dialed down until it vanishes. Blue that was always underneath, hidden beneath the melanin, becomes visible for the first time. In a brown iris, melanin, 93 to 95% of the end anterior stromal surface. In a blue iris, that coverage drops to less than 5%, sometimes functionally zero. The difference is not a qualitative shift. It is a collapse in melanin density measurable under any microscope. Structural blue was always there. Every human iris, even the darkest brown, has the collagen mesh that scatters short wavelengths. The scattering still happens. But in a brown eye, the thick melanin absorbs that scattered blue light before it can escape the iris and reach an observer's retina. The blue is produced and then swallowed, invisible. The mutation simply removed the absorbing layer and let the pre-existing blue leak outward. This is why a blue-eyed child can be born to two brown-eyed parents who each carry one copy of the RS12913-832 variant. The child inherits the same OCA2 gene, largely intact, but now the regulatory switch near HERC2 instructs that gene to operate at a whisper instead, a shout. The melanin production pathway still exists. It has simply been instructed by a single nucleotide change in a patch of non-coding DNA to run at a fraction of its capacity. The blue eye is a brown eye with less of the thing that makes it brown. An absence, not a creation, abstraction that revealed what was hidden. and this. Subtraction happened exactly once. A single molecular accident in the enhancer of a single gene, in a single cell, in a single developing iris. One nucleotide, one human being, one moment. And the echo, 10,000 years later, is the gaze of roughly 600 million living people. In Copenhagen, inside a quiet laboratory at the University of Copenhagen, a geneticist named Hans Eiberg was staring down a question that, on its surface, sounded almost too elementary to ask. The year was 2008, and the paper would be published in Human Genetics, and it would overturn a quiet, unspoken assumption that had hung around the subject for as long as anyone had wondered about it. The colour of human eyes. Eiberg's team wanted to know whether the mutation that turned an eye blue was the same mutation. The same microscopic spelling error in every person possessed it. Or had nature stumbled onto the same trick more than once in more than one place across the sprawling history of Homo sapiens? The answer, when it arrived, left no question open. Eiberg and his colleagues collected DNA from 155 living individuals, every one of them with blue eyes. They were not a family group, they did not share a village, a dialect, or a recent ancestry. The samples were drawn deliberately from across the geography of Europe, from the coasts of the Baltic, from the hills of the Iberian Peninsula, from Ireland, from the fjords of Scandinavia, from the plains of Hungary. The point was to maximize diversity of lineage, to ensure that if the mutation had multiple origins, those separate histories would reveal themselves in the genetic data. Then the team went to work. They genotyped each of those 155 blue-eyed individuals across 842,000. Single nucleotide polymorphisms. A SNP, single nucleotide polymorphism, is nothing more than a point in the genome where one person's DNA might read adenine while another's reads guanine. One letter out of the more than 3 billion chemical letters that make up a human being. Across 842,000 such positions. The researchers scanned the genomes of these unrelated people, searching for a shared signal, a region where all 155 blue-eyed individuals carried the same genetic text, while brown-eyed individuals did not. They found it on chromosome 15. The specific variant was RS129138832. An adenine that had become a guanine sat it not inside the protein-coding portion of a gene, but in a regulatory stretch of the HERT2 gene, a segment that functions as a dimmer switch, controlling the expression of the neighbouring AOCO gene. CA2 is the gene that drives melanin production in the iris. The variant in HERX2, the team confirmed, binds regulatory factors differently, dialing OC2 expression downward. And in every single one of the 155 blue-eyed individuals, everyone, without a single exception, the adenine to guanine change was there, identical. The same base pair at the same chromosomal address in every sample, but the identity did not end at the single letter. If a mutation originates once, the surrounding DNA gets inherited along with it, only the slow churn of recombination. The crossing over of chromosomes during reproduction gradually breaks that ancestral block into shorter pieces, generations. A young mutation, one that has existed for only a limited number of generations, will still be embedded in a long, continuous stretch of original sequence, not yet chopped up by the shuffling of the ages. An old mutation, by contrast, will sit in a short, scattered segment, its original surroundings long since dispersed by recombination across countless ancestors. What Iberg's team measured next was the length of that surrounding block in the blue-eyed individuals. They looked at the DNA on either side of RS129-13832 on the same chromosome and asked how far the conserved haplotype extended. How much of the original ancestor's genetic neighbourhood was still travelling with the variant? The number they arrived at was stunning. Roughly 166 kilobases, or 166,000 base pairs, largely unshuffled DNA. And this was not a feature of one subpopulation alone: the Dane, the Spaniard, the Irish woman, the Estonian. All of them carried this same long, unbroken block of sequence, their chromosomes unrecombined across the entire stretch. That length, 166,000 base pairs, is a direct form of clock. In human populations, recombination acts on average about once every 100 million base pairs per generation, though the rate varies across the genome. To have a haplotype block of that size remain intact across unrelated individuals living in opposite corners of a continent is a signal that the block has existed for only a few hundred generations. If the mutation were old, say 200,000 years old, dating to the early days of our species, recombination would have long since ground the surrounding DNA into fragments of a few kilobases or less. The intact block was a certificate of youth. The team calculated the probable age, using models that account for recombination rates in this part and this part of the genome, and the central estimate settled near 10,000 years, plausible range from about 6,000 to 12,000. On an evolutionary timescale, that is almost nothing, is the genetic equivalent of yesterday. This was not a result that required elaborate statistical defense or paragraphs of caveats. 155 unrelated individuals, 842,000 markers, and the signal at RS129-13832 was monolithic, one exception. Not one blue-eyed person carrying a different variant, not one instance of the variant sitting on a different haplotype background. The data were so clean that the conclusion was inescapable. Iberg himself, speaking to journalists after the paper's publication, distilled it into two sentences. The first, originally, we all had brown eyes, but a genetic mutation affecting the OCA2 gene in our chromosomes resulted in the creation of a switch that literally turned off the ability to produce brown eyes. The word literally is not casual. The enhancer region in HERC2, where RS129-13832 sits, had lost its capacity to drive normal OCA2 expression. The genetic switch had been thrown, and it had stayed thrown, in every descendant who inherited two broken copies. He added that this was a clear example of how a simple genetic change, a single mutation, can have a profound effect on human appearance and diversity. One nucleotide, one human being, one moment, and the echo, 10,000 years later, is the gaze of roughly 600 million living people. The data left no room for independent origins. If the adenine to guanine change had occurred more than once in different populations at different times, the surrounding haplotypes would differ. They did not. The long block of conserved DNA was uniform across the entire sample, across the entire continent. The parsimonious, indeed the only, explanation was a single common ancestor. One developing embryo, one cell, one unrepeated chemical accident that inserted a guanine where evolution had placed an adenine in every human before. Blue Eye, alive today, is a copy, transmitted and retransmitted through 400 generations of human descent. 10,000 years. The number comes from the silent arithmetic of recombination, the way a mother's chromosome and a father's chromosome swap sections during the formation of an egg or a sperm, every generation without fail. Each shuffle cuts and reassembles the long blocks of ancestral DNA. The longer the unbroken sequence around a mutation, the fewer generations of shuffling have occurred, and the shorter the time since that mutation first appeared. When the Copenhagen team sequenced the blue-eyed individuals in their 2008 study, they found that the RS129-13832 allele sat at the centre of a haplotype block spanning roughly 166,000 base pairs of DNA that had not yet been disrupted by recombination. That length is a stopwatch. Run the clock backward at the average human recombination rate, and you arrive at a central estimate of about 10,000 years, bracketed by a range from roughly 6,000 to 12,000 years. On a geological scale, that is less than a breath. On a human evolutionary scale, it is almost the present moment. Place that figure against the full span of Homo sapiens. Our species emerged in Africa about 300,000 years ago. First, 290,000 years. More than 95% of our existence. Every living human eye on this planet was brown, blue eyes, measured from their first flicker to this evening's sunset. Occupy at most the final 3 to 5% of the human story. The oldest cave paintings at Lascaux are 17,000 years old. The last Neanderthals vanished roughly 40,000 years ago. First blue-eyed child drew breath well after all of that. Separated from the age of cave bears and ice sheets by only a thin margin of millennia. The world that child was born into was not the Europe of cathedrals and wheat fields. 10,000 years ago, the last ice age had only recently loosened its grip. Great ice sheets were still bleeding fresh water into the oceans, and sea levels were climbing rapidly, redrawing coastlines every human lifetime. The Black Sea, at that moment, was a dramatically different body of water. Lower, smaller, its basin still being reshaped by the returning Mediterranean through the Bosporus. People living in southern Europe and around the Black Sea littoral were Black Sea Zimayek, Nunglavik's hunter-gatherers. They moved in small, mobile bands of 20 to 50 individuals, following red deer and wild boar, harvesting hazelnuts and wild grains, reading the landscape with an intimacy modern humans have almost entirely lost. Their toolkits were microliths, tiny, geometrically precise blades of flint set into wooden shafts, and they buried their dead with ochre and pierced shells. They did not farm. Agriculture had begun in the Levant, in the Fertile Crescent, perhaps 2,000 years earlier, but it had not yet spread in any substantial way across the European landmass. No permanent villages through most of the continent, fired clay pots in the earliest phases of this period. No writing, no metal, no wheel. The first blue-eyed human lived in a world that, technologically and demographically, was close to the Paleolithic than to the Bronze Age. Measured against other milestones of human prehistory, Mutation is startlingly young. The domestication of the dog reaches back at least 15,000 years, the oldest known dog burial at Bonn Oberkassel in Germany, dated to about 14,000 years ago. The oldest known pottery is roughly 20,000 years old. Stonehenge was begun around 5,000 years ago. Blue Eye Mutation is at least 5,000 years older than Stonehenge, and by the time those first stones were raised on Salisbury Plain, blue eyes had already been present in Europe for as long as Stonehenge has stood to the present day. Trent is roughly twice as old as the pyramids of Giza, which rose about 4,500 years ago. If you could stand in a Mesolithic camp 10,000 years ago and gaze into the eyes of that first child, be looking at a feature that would not appear on a pharaoh's face for another 5,000 years. There is a further layer to the clock, and it is written in the rules of inheritance. The RS1913832 variant is recessive. An individual must inherit 2 copies to have blue eyes. Person with 1 mutant copy and 1 standard brown eye allele will have brown eyes, carrying the mutation entirely invisibly. A heterozygote, silent vessel. The allele could move through populations for generations without ever manifesting. Band of Mesolithic foragers might have contained dozens of carriers, all brown-eyed, all unremarkable, while the variant passed silently from mother to daughter, from father to son, waiting for the moment when 2 carriers produced a child together. Imagine a woman somewhere near the Danube Delta or the northern shore of the Black Sea, holding her newborn to the light and seeing, for the first time in any human lineage that would survive, an iris that reflected the sky rather than the earth. She had no word for it. No one in her band had ever seen such a thing. The child's eyes were not a sign, not a gift, not a curse, just a curiosity, a small biological surprise that meant nothing at all until that child grew and had children of her own. Timing of that eruption places it squarely at a hinge in prehistory. 10,000 years ago, the climate was warming, forests were spreading northward across Europe, and the pattern of human life was beginning a slow, uneven pivot from mobility towards settlement. In the Black Sea region, the changing geography would have reshaped the territories available to hunting bands. Populations were likely small, fragmented, and subject to the founder effects that amplify rare alleles by chance. In that ecological pressure cooker, recessive mutation, with no measurable survival advantage, was in a position to catch hold and, gradually, to spread. First blue-eyed child may have been a complete surprise to her community, visible eruption of a hidden genetic current that had been flowing beneath the surface for centuries. The mutation appeared, to understanding where it first appeared. On what coastline, in what river valley, under what particular sky, is a different order of problem. The genetic stopwatch narrows the search to a handful of centuries in the Mesolithic. The location has so far resisted every effort to pin it down. The 2008 Copenhagen study did not claim to know. It proposed a region, the Black Sea Basin, the northern Mediterranean, Caucasus Corridor, based on the distribution of the RS0129133832 allele in modern populations, and on what we understand about the migration routes that carried Mesolithic hunter-gatherers across the European continent. That logic pointed south and east toward the land bridge between Anatolia and the Balkans, toward the shores of a Black Sea that was still filling from glacial melt, toward a landscape of sheltered river valleys and coastal plains among that would have supported exactly the kind of small, mobile band the genetics implies. But distribution data is not proof. It is a fingerprint on a window, not a photograph of the hand. No ancient DNA from the critical time and place has been recovered and tested for Rs129133832. The window we need is roughly 10,000 years ago. Place. The place is somewhere within a region spanning perhaps 1,000 kilometers of coastline and inland steppe. Not a single skeleton from that exact conjunction of time and territory has yielded a genome that can be screened for the mutation. The science of ancient DNA is fast, brilliant, and relentlessly constrained by preservation conditions. Bone survives where chemistry allows it to survive, and the acidic soils of the Black Sea littoral are not generous to collagen, so the birthplace remains contested, and 3 geographic hypotheses sit on the table, each with its own logic, each with its own silences. Black Sea corridor hypothesis remains the consensus default. It draws strength from the modern frequency gradient and from archaeological part, evidence that the region was a crossroads for human populations during the Mesolithic. A place where bands moving north from the Near East and west from the Eurasian steppe would have met, mixed, and carried new genetic variants onward into the European interior. The Danube Basin, just upstream from the Black Sea, was one of the primary highways by which people entered Central Europe after the ice retreated. A mutation arising anywhere along that corridor would have had access to an entire continent. Then there is the Iberian challenge. In 2014, when the Lobrania genome was sequenced, the blue eye allele appeared 7,000 years ago in the mountains of northwest Spain. That is not old enough to represent the founder. The founder lived earlier, but it is old enough to make the question uncomfortable. If the mutation was already in Iberia 7 millennia ago, and the estimated origin date is between 6,000 and 10,000 years before present, then the founder either lived quite close to Lobrania's era, or the mutation spread westward far faster than the gradual diffusion model assumes. Some researchers have asked whether the Iberian Peninsula itself could be the birthplace. A refuge population, a host, isolated during the last glacial maximum, expanding northward as the ice sheets withdrew, carrying a neutral mutation that had arisen locally. The objection is that modern Iberian populations do not show the extremely high blue-eye frequencies we see in the Baltic. But modern frequencies are not ancient ones, and population replacements during the Neolithic and Bronze Age reshuffled the genetic map of the peninsula more than once. Third possibility sits to the north. Scandinavia. The argument here is not distribution-based, but adaptive. In the extreme low-light conditions of a northern winter, when daylight contracts to a few hours of twilight, an iris with reduced melanin allows slightly more light to reach the retina. That slight advantage, compounded over thousands of generations, might have been enough to favour a mutation that arose locally or arrived with the earliest post-glacial colonists. The Lobrania skeleton, with its dark skin and blue eyes, shows that the mutation did not require a northern latitude to exist. But it might have required one to thrive. Problem is timing. Scandinavia was under ice until roughly 11,000 years ago, and the first human reoccupation was slow, patchy, and archaeologically thin. A mutation appearing in those first small bands would have had a vanishingly shallow pool of potential carriers, Black Sea. With its denser networks of interconnected groups, offers a more plausible demographic engine. Each hypothesis has its defenders and its gaps, and that is the honest counting. The exact latitude where the first blue-eyed child sat by her fire, the coastline she might have walked, the language her mother spoke, the burial customs her band practiced. All of it is unknown and may remain so. This is not a failure of science. It is the precise shape of what is currently knowable, drawn with the same care as the known parts. The edge of the map is marked honestly. What would change that? A Skeleton, a single well-preserved individual from roughly 10,000 years ago, found in a specific region, carrying the RS1291383 allele in an ancestral haplotype context that matches the modern version. If that skeleton turned up in a sinkhole in the Caucasus, the Black Sea hypothesis firms into something close to settled. If it turned up in a cave in Cantabria, the Iberian argument strengthens. If it turned up on the Baltic coast, the northern case gains ground. The ancient DNA community is searching. The tools are faster than they were in 1008, and the sample libraries are growing every year. The dirt yields its dead on its own schedule, and the find that would lock the birthplace into place has not yet been made, and if she lived, then the mutation did not stay there. It moved. In 2014, a genome emerged from a cave in the Cantabrian mountains that looked nothing like the story European prehistory had been telling itself. The cave is called Labrania Arintero, a limestone shelter near the village of Arintero in the province of Leon, northwest Spain. Caving enthusiasts had known the site since the 1980s, but it took archaeologists until 2006 to begin excavating its deep, dry deposits. In that chill, organic matter holds its shape for millennia. Two male skeletons rested there, side by side, placed in the cave floor without stone tools or ornaments, only the naked bone. One of those skeletons, Labrania 1, was remarkably intact. Collagen survived in the tooth roots. DNA survived. Toast. And when a team from the Institute of Evolutionary Biology in Barcelona sequenced the entire genome in 2014, recovered a face that upended every quiet assumption about what ancient Europeans looked like. The team was led by Inigo Olalde and Carles Laluza Fox. They extracted DNA from the root of a lower molar belonging to a man who had been dead for 7,000 years. The radiocarbon calibration placed his death around 5,000 BCE, give or take a century, firmly in the late Mesolithic, that date. Agriculture had not yet reached the Cantabrian valleys. This man lived by hunting and foraging, moving with the seasons through oak and beech forests that covered the limestone ridges broad. The genome that came back from the sequencer has carried more than a dozen ancestry markers. But the pigmentation genes hit the team first. They ran the predictive panel for the OCA to HERT2 region. The results stopped them. This Mesolithic hunter-gatherer had blue eyes, 2 copies of the RS1291383 allele, homozygotus for the loss-of-function variant that dials down OCA2 expression to near 0 in the iris stroma. He also had dark skin. The SLC24A5 and SLC45A2 alleles that later lightened European skin were entirely absent. His skin would have been a deep brown, adapted to the high ultraviolet exposure his ancestors experienced for tens of thousands of years across the Near East and Africa. Before entering Europe, his hair was dark, near black. The Kittil, Poo, and other later mutations that produced blonde and red hair had not yet appeared in his lineage. That combination, pale blue eyes set against brown skin and black hair, is almost vanishingly rare in modern Europeans. Branya 1 is the earliest direct physical confirmation that the RS12913832 mutation existed in a real human being at least 7,000 years ago. The genetic readout was unambiguous. The HiRIS-Plex forensic pigmentation prediction system returned a probability exceeding 0.99 for blue eyes, a 0.94 probability for dark skin, and a 0.98 probability for dark hair. He is not a statistical inference. He is a specific man who walked the steep-sided valleys of what is now Leon. He hunted ibex and red deer with microlith-tipped spears. He stood about 1.70 m, lean, his bones showing strong muscle insertions from a life of movement. When he died in his mid-30s, community laid him in the cave, without objects, without ornament, without any mark that would tell us his name or rank, and his eyes were blue. Discovery shatters a quiet linkage that many of us carry without thinking. We tend to bundle light eyes, light skin, light hair into a single ancestral story. Labrania proves the package never existed as a unit. Blue eyes came first, Pgruk, arriving by a regulatory mutation in a single individual, spreading among dark-skinned, dark-haired hunter-gatherers across wide distances. Light skin came later, in a separate adaptation. The derived alleles of SLC245 and SLC45A2 swept through European populations only with the arrival. Neolithic farmers from Anatolia around 7,000 to 6,000 years ago in the east, and even later in the Iberian Peninsula, farming populations carried lighter skin as a genetic signature, already ascending under selection for vitamin D synthesis in low sunlight environments. Blonde hair arrived even later than that. The Kittel variant associated with blondness began sweeping through northern Europe during the Bronze Age, thousands of years after the blue eye mutation was already in motion. Three traits we now associate with one another are independent evolutionary events that happened to converge in certain regions of Europe over a span of 5 to 7 millennia. Not a single makeover, but a staggered assembly of accidents, each with its own timing, its own geography, own selective story. Dark skin and blue eyes is not a paradox. It is simply what the RS12913832 mutation looked like before the other mutations arrived. The Osea HER2 switch only acts on the melanin-producing cells in the iris. It does not touch the melanin in the skin. The skin-lightening alleles that dominate modern Europeans had not yet entered the Labrania man's genome in any form. Skin remained dark, default state, for a species that spent most of its evolutionary history under an equatorial sun. He is not an anomaly. He is a snapshot of the genetic landscape in the middle of a long, complicated process that was nowhere near finished. Man from Labrania forces another calibration. The timeline tightens. Linkage disequilibrium analyses of modern blue-eyed chromosomes place the founder mutation between 6,000 and 10,000 years before present, the central estimate near 10,000 years. If the RS12913832 allele was already in northern Spain when Labrania 1 died around 7,000 years ago. Then the founder lived very close to him, perhaps only 2 or 3 centuries earlier. The mutation was still young, still rare, still silent in many carriers who passed it as a heterozygote without ever seeing it expressed. And it was already moving. It had travelled from whatever parent population first sheltered it to the limestone highlands of the Cantabrian coast, threading through networks of small hunter-gatherer bands along routes we can only guess at. Now we had a real face on the map, not the founder. That child by the fire remains irretrievable, but a named archaeological individual who proves the mutation was not an abstraction confined to a computer model. He was a man who saw the world through pale blue irises, under dark brows, against dark skin, in a landscape that knew only firelight and starlight. For 7,000 years his bones waited in the cold dark, waiting for scientists to pull a tooth and read the single switched letter that told them. His eyes were pale. That confirmation only sharpened the next question. If the mutation was already in southern Europe 7 millennia ago, how did it eventually reach near fixation in Estonia and Finland? 89% frequency, while dwindling to single-digit percentages across the very Iberian Peninsula where Labrania lies buried. The map now had a fixed point, a real face, but the mystery of how that face became the face of Northern Europe had only grown. From one pair of pale eyes to hundreds of millions, the number is not a metaphor. Roughly 8 to 10% of every human being alive on Earth today carries 2 copies of the RS129-13832 allele, between 600 million and 800 million people. All of them descend from that single Mesolithic founder, the child, whose face we cannot see and whose name we will never speak. The mutation travelled farther than any empire built in the intervening millennia. Look at the modern map and the pattern is stark. Blue eyes are not evenly distributed across the world. They cluster tightly in northern and western Europe, thinning rapidly as you move south and east. Estonia, 89% of the population has blue eyes. Finland, 89%. In those 2 nations on the Baltic Rim, brown eyes are the rarity, visible minority. Nearby, Iceland reaches 75%. Ireland and Scotland each register 57%. Denmark sits at 55%. These are population-level frequencies. Whole countries where the majority of people carry the same recessive allele in double copy. The same loss-of-function switch turned off in the same gene. In the same position, changing the colour of the sky in every second face. Move away from that northern European core, and the frequencies drop fast. Germany and the Netherlands sit lower. Southern Europe, Italy, Greece, the Balkans, falls to 20%, 15%, single digits in places. North Africa and the Near East hold small pockets, remnants of migration and trade across the Mediterranean, and the Levant over thousands of years. Farther east, blue eyes appear occasionally in Central Asian populations at frequencies that never exceed a few percent. The ancestral heartland is unmistakable. The cold northern rim of Europe, where winter daylight is thin and the sky is grey more often than blue. The allele moved with human bodies across a landscape. It followed the routes of Mesolithic hunter-gatherers, fanning out from whatever origin point first sheltered the mutation, threading westward along the river valleys and coastlines of a Europe. still half wild. Later migration waves carried it further. Neolithic farmers entering from Anatolia after 7,000 years ago brought their own genetic signatures, lighter skin among them, and some of those farmers already carried the RS 129-13832 variant as heterozygotes, Bronze Age pastoralists from the steppe. The Yamnaya-related populations, whose expansion reshaped the genetic landscape of Europe around 5,000 years ago, carried the allele north and west at frequencies that seeded the high concentrations we see today. The trait was never static. Post, it moved. Whenever people moved, with bands of hunter-gatherers following reindeer herds, with farmers clearing forest for wheat, herders driving cattle across open grassland, with every marriage, every birth, every journey that connected one river system to the next. But the numbers pose a problem that simple migration cannot solve. For a recessive trait to reach 89% frequency in Estonia in roughly 400 generations demands a force stronger than neutral shuffling. Baseline expectation for a mutation like RS 129133832, a regulatory change that reduces melanin in the iris and offers no measurable survival advantage, is that it should remain rare. Most neutral mutations do. They appear, they drift at low frequencies for a while, and most eventually vanish. This one did not vanish. It rose from a single copy in a single individual to near fixation in multiple populations in a geographic blink. The mathematics are unforgiving. Either the selection pressure driving it was extremely strong, populations that carried it passed through extreme bottlenecks, where chance alone amplified the allele to high frequency. Both forces acted in combination over the 10 millennia since the founder lived. There is no fourth option. The inheritance pattern makes the climb even steeper. Because the trait is recessive, it can travel invisibly through generations. Heterozygote. One copy of the mutation, one copy of the ancestral brown eye allele has brown eyes. That person can live an entire life, children, grow old, and die without anyone knowing. The blue eye switch sits silent in their genome. Only when two carriers reproduce together does the trait surface in roughly 1/4 of their children. For much of the allele's early history must have moved like this, hidden in the genomes of brown-eyed people, surfacing rarely, a startling flash of pale iris in a dark-eyed community, then vanishing again. For generations, if the blue-eyed child did not survive or did not reproduce, the speed of the spread, from 0 to nearly 90% in 400 generations, was faster than the visible blue-eyed children alone would suggest. The hidden carriers did the work, generation after generation, threading the mutation through populations that never saw it expressed, one more number to hold. The founder lived roughly 10,000 years ago, past, and the Labrania man carried the mutation in double copy 7,000 years ago. That leaves the entire climb from a handful of carriers, as in Mesolithic Spain, to near fixation in modern Estonia, to happen in the last 7 millennia. Less, if the highest frequencies were reached only in the last 2,000 or 3,000 years. The allele did not inch upward at a steady rate, it surged, and it did so in the absence of any confirmed survival advantage. The RS 129133832 allele does not protect against UV damage, does not improve night vision. It does not confer resistance to any known pathogen. As far as the best current biology can determine, it does nothing at all, except change the colour of the eyes in daylight, which leaves the question hanging, sharp and unresolved. What combination of forces, pushed a biologically neutral accident from one child to roughly every second face on the streets of Helsinki? Host. The next chapter lays out the three leading explanations, each with its own strengths and its own silences. First explanation is the one most people reach for instinctively: sexual selection. Blue eyes are rare. They are highly visible. In a world where every human iris was brown, a pair of pale blue eyes would have drawn immediate attention, if that attention translated into greater reproductive success. If the first blue-eyed individuals were perceived as more attractive, more memorable, more desirable as mates, then the allele could have risen in frequency, generation by generation, not because it conferred survival, because it conferred courtship. The mechanism is well understood in evolutionary biology. Peacock's tails, birdsong, the elaborate displays of bowerbirds. None of these help an animal live longer. They help it reproduce. A conspicuous, harmless mutation like blue eyes fits that pattern neatly. And in a small Mesolithic band of a few dozen people, where every face was known and every social bond was continually renegotiated, small difference in perceived attractiveness could have an outsized demographic effect over time. The limit of the sexual selection hypothesis is not that it is implausible, it is that it is untestable. No archaeological record preserves mate preference from 10,000 years ago. No cave painting shows who was courted and who was ignored. Psychologists today have attempted to measure whether blue eyes are perceived as more attractive, and the results are inconsistent, culturally contingent, and impossible to extrapolate backward into a world of mobile hunter-gatherers whose social rules we do not know. We cannot reconstruct the gaze of a Mesolithic parent noticing a blue-eyed child, or the reaction of a potential partner meeting that child grown, because the human brain that formed those judgments left no trace. The selection may have been sexual. It may have been strong, probably never know. The second explanation shifts the focus from the trait itself to its genetic neighbourhood. Mutation sits in a regulatory region of the HERS2 gene on chromosome 15, and that region is physically close to genes that control skin pigmentation, notably SLC24A5 and SLC45A2, both of which were under strong positive selection in European populations as humans moved into low sunlight latitudes and needed lighter skin to synthesize sufficient vitamin D. The blue eye allele may not have been the target of selection at all. It may have been a passenger, hitchhiking alongside a nearby mutation that was genuinely beneficial for survival. As natural selection drove lighter skin to higher and higher frequency across Europe, the Rs1129133832 variant may have simply been dragged upward with it, trapped on the same stretch of chromosome, unable to recombine away fast enough to escape the ride. Geneticists call this linkage, and it is a perfectly ordinary phenomenon. The product is a trait that looks selected, but is merely linked to something that is. The problem with the hitchhiking hypothesis wears a face: Branya. The 7,000-year-old hunter-gatherer from the mountains of northwest Spain carried the blue eye mutation in double copy, and his skin dark. He had the same allele that 500 million people carry today. a cost, and he did not have the lighter skin that the hitchhiking model requires as the engine of its spread. If the blue-eye allele travelled exclusively alongside lighter skin, then the two traits should appear together in ancient genomes. They do not. The decoupling is undeniable. Dark skin and blue eyes coexisted in that single individual, and almost certainly in many others of his era. The hitchhiking model is not disproven by this. Skin lightening alleles may have been selected later, or in different populations, blue eye allele may have come along for part of the ride. But the clean picture of a single linked block sweeping to fixation across the continent collapses under the weight of Labrania's genome. The two traits were travelling separately, or they became coupled late. Timing is unresolved. The third explanation is the simplest, and in many ways the most honest. Random genetic drift. In small, isolated populations, the frequencies of neutral mutations do not follow a predictable path. They fluctuate. They rise and fall in a statistical dance that depends on who lives, who dies, who migrates, and who happens to have children. Mutation, like RSRS12913832, could have drifted upward, simply because the band that carried it was in the right place at the right time, and because the descendants of that band founded larger populations as the glacier populations as the glaciers retreated, and Europe filled with people again. Founder effect at the northern edge of the habitable world, where group sizes were small and the gene pool was narrow, pushed the allele to high frequency in a few dozen generations. No selection required. No mating preference required. Just the stochastic arithmetic of finite populations. Drift alone, however, strains to explain the modern frequencies. A neutral mutation in population of hunter-gatherers, as numbering and left randomly, would drift, often to extinction, rarely to intermediate frequencies. But it would not, by chance alone, reach 89% in Estonia and 75% in Iceland, while remaining at 2% in Afghanistan, unless the founding populations that settled those northern regions were so tiny that the random sampling extreme, and unless later population growth was explosive and continuous without diluting gene flow from outside. Drift is a necessary piece of the puzzle. It must have occurred, but by itself, it leaves a gap between what the mathematics of neutral evolution predicts and what the map of modern eye colour shows. Extreme bottlenecks can help. Population models that combine drift with moderate selection can help more. But drift alone without any assistance struggles to make the numbers work. The honest synthesis held by most geneticists who work on this problem, that no single force carried blue eyes from a child by a fire to 500 million people. Sexual selection may have amplified a trait that was noticed and valued. Hitchhiking may have pulled the allele along for stretches of the journey, particularly as lighter skin became common in later millennia. Drift may have locked in high frequencies in the small, saturated gene pools of far northern Europe after the initial spread. The relative weight of each mechanism is genuinely unresolved, not because science has failed, but because the evidence, the precise mating decisions of people who lived 10,000 years ago, the exact selection coefficients on individual mutations, the demographic sizes of every Mesolithic band, is beyond the reach of any method we currently possess. This is not a failure of explanation. It is the shape of the problem itself. The data that would resolve it are incomplete. False. The models that could weigh the forces against each other produce a family of plausible scenarios rather than a single victor. What remains is a question that sits squarely in the open. Why did this one neutral accident become the signature of a continent's eyes? She has no body, no grave goods, no ochre-stained burial, no carved figurine left beside her head. Not a single tooth survives. Nothing that an archaeologist can label, photograph, or place behind museum glass. She is genetically certain and archaeologically non-existent. A person deduced from the DNA of living strangers, not recovered from the earth that held her. The gap between those two kinds of knowledge is the subject of this chapter. If you walked the coastline linear where she likely lived, somewhere near the Black Sea, perhaps, or the northern Mediterranean, the location still unresolved, you would not find a marker. There is no mound, no midden that can be tied to her hands, cave wall that records her face. The soil of 10,000 years has buried and reworked and erased the surface she walked on. Sea levels have risen. Rivers have shifted. The campsite where she sat by the fire is under water, or under a modern city, scattered into anonymity by 10 millennia of wind and rain. The Mesolithic left us flint blades and hearthstones and the occasional partial skeleton, but it did not leave us her, and yet she existed. The genetic evidence is not ambiguous on this point. The identical RS129138832 haplotype shared by every blue-eyed person tested. The same switch, in the same position, on the same chromosomal background, cannot have arisen independently in multiple surviving lineages. It is too specific. The surrounding haplotype block is too long, too unshuffled by recombination. If separate blue-eye mutations had occurred in separate people, and both lines had survived, the haplotype backgrounds would differ. They do not. The entire living population of blue-eyed humans converges on a single molecular event, in a single individual, at a single moment. Mathematics of population genetics is not sentimental, but on this one point it is absolute. She was real. What the mathematics does not reveal is whether she was the first. The mutation may have occurred earlier, may have appeared in an individual who lived 12,000 years ago, or 15 or 20, or 2, someone who carried it, passed it to children, and whose lineage then ended. A fever, a bad winter, a conflict with a neighbouring band, the extinction of a small family group in a world where the average life expectancy was 30 years, and the margin for error was 0. That happened. The earlier lineage left no descendants, and the RS129138832 we see in the modern world descends from a later occurrence, the one that survived. The date we have, 6 to 10,000 years ago, is the date of the surviving founder. It is not necessarily the date the mutation first flickered into existence in a human genome. It is the date of the lineage that made it through. This is not a weakness of the science. It is a precise description of what lineage survival means. Mutations arise constantly. Most are lost. The ones we see are the ones that did not go extinct. A sample biased by survival. Not a record of every attempt. Blue-eye mutation may have been tried more than once. The one that stuck is the one we carry. What did her community see when they looked at her? The question is unanswerable in any rigorous sense, but it is not therefore meaningless. In a band of 30 to 50 people, every face is known, every variation in appearance is noticed. a child with pale blue eyes. The only such eyes in her world, the only such eyes in any world anyone alive had ever seen. Would have been visually singular in a way that is difficult to reconstruct now. Blue eyes are common across entire nations. There was no word for the colour, no category, cultural precedent for what her irises did to the light. The reaction of her family, her peers, any potential mates later in her life is not recorded in any surviving material. Did they see it as beautiful, strange? A sign of something? Did they treat her differently? And did that treatment help or hinder her chances of survival and reproduction? We do not know. We cannot know. What we can say is that she passed the allele on. How many children she had is unknown. The lineage could have bottlenecked through a single child, single grandchild, a single carrier who nearly died before reproducing. The fact that the haplotype is uniform across all blue-eyed people suggests a tight bottleneck, a small number of carriers in the early generations, perhaps only one or two, from whom all subsequent copies descend. The mutation survived. But it may have come very close to vanishing. Iberg and his team, working in their laboratory in Copenhagen, did not set out to find a ghost. They set out to map the genetics of human eye colour. What they found instead was a single person staring back at them through the data. A person whose existence was mathematically inescapable, but whose name, sex, language, and burial location the data could not supply. The moment in the lab when the haplotype uniformity became clear must have been strange. The realization that every blue-eyed donor in the study, regardless of nationality or background, tracing back to the same molecular accident. And yet, no name. The most connected person in human history, linked genetically, directly, to 600 million living people, is entirely unknown. No monument marks her. No song remembers her. The only record of her existence is the nucleotide sequence in the cells of her descendants, text she could not read and did not know she was writing. She did not know she was writing anything at all. That is part of the weight. Mutation was not a decision. It was a copying error during cell division. A single nucleotide substituted for another in a regulatory region of a gene she had never heard of. In a body she inhabited without understanding, a world where the concept of a gene did not exist and would not exist for another 10,000 years. She lived her life unaware that her eyes were the first of anything. She may have died before any of her children showed the trait. If the recessive inheritance pattern held, the blue vanished for a generation, hidden in heterozygotes, only to reappear in a grandchild whose eyes caught the firelight the same way hers had. And someone in the band noticed, and the story of the eyes began to move through time. The dignity of not knowing her name is not a concession. It is an honest boundary. She remains a placeholder for a person we will never recover. A narrative device that respects the gap between what the DNA tells us and what the has chosen to keep. That gap is not a failure. It is the precise shape of what is currently knowable, and it will not be closed by better sequencing or larger sample sizes or more powerful computers. Founder's body may someday be found, a skeleton from the right place and the right time, carrying the RRS 129138332 mutation in homozygous form, its genome placing it at the root of the haplotype tree. That would be a revelation. It would give us bones. It would give us a location. Would not give us her name. It would not tell us what her mother said when she first looked into her eyes, or whether she was loved or feared, simply accepted as another child in a band that had other things to worry about. Food, shelter, the coming winter, the memory of the last one. Some things the genome cannot encode. Some things are lost because they were never stored in any physical medium at all. She is the ghost at the centre of this story. A point of origin that the science proves but cannot touch. And if you sit with that long enough, forces a question that the genetics opens, but is not designed to close. What does it mean that so much of what we look like began with accidents? This specific, this recent, and this close to being erased. Tacitus wrote it down. In 98 CE, the Roman historian published Germania, his ethnographic portrait of the tribes beyond the Rhine. Among the physical descriptions he recorded was a detail that has echoed for 2,000 years. He wrote of fierce and blue eyes. He was not describing an anomaly. He was describing a population-level characteristic, common enough among the Germanic peoples that a Roman observer considered it worth noting for his audience back in the Mediterranean world. Brown eyes were the overwhelming norm. By the time Tacitus wrote those words, the blue eye mutation was already old. 8,000 years old, at minimum. It had moved across Europe through routes that no Roman historian knew existed, carried in the genomes of people who had no writing, and left no ethnographies of their own. Romans gave it a name and a tribal association, Germanic, and in doing so, they began the long cultural process of attaching meaning to a molecular accident that had none. The cultural mythology of blue eyes as an ancient, defining northern trait was born roughly 2,000 years ago. The trait itself is at least 4 times older than the mythology. That gap between the genetic reality and the story we started telling about it is the subject of this chapter. By the time Rome was an empire, blue eyes were well established across northern and western Europe. But their climb to that visibility had taken most of the preceding 8 millennia, and it did not follow a single straight path. The ancient DNA recovered in the last decade has revealed a complex, multi-stage spread that maps onto the major population transformations of European prehistory. First stage was the Mesolithic hunter-gatherer world. The Labrania individual in Spain 7,000 years ago proves the allele was already present in Western Europe by that date. He was not alone. Ancient genomes from Mesolithic sites have since yielded additional carriers of RS12913832, not as the majority type, but as a persisting minority variant moving through the interconnected bands that ranged across the post-Ice Age landscape. These were small populations. The allele could have easily been lost. It was not. The second stage came with the Neolithic transition, beginning around 7,000 to 6,000 years ago in southeastern Europe, and spreading northwest over the following millennia. This was not a simple replacement of hunter-gatherers by farmers. Ancient DNA has shown that the first agriculturalists arriving from Anatolia carried predominantly brown eyes. But as farming communities expanded and interacted with existing hunter-gatherer groups, gene flow moved in both directions. The RS12913832 allele crossed the contact zone. It appears in Neolithic genomes from Hungary at roughly 7,000 years ago, from Germany 1,000 years later, from Sweden by the Middle Neolithic. It moved with people, with marriages, with alliances, with the slow incorporation of hunter-gatherer lineages into farming settlements. But it did not sweep. It remained a minority variant, present at low to moderate frequency for thousands of years. The third stage arrived with the Bronze Age, starting roughly 5,000 years ago, when a massive expansion of pastoralists from the Pontic Caspian steppe rolled westward into Europe. These Yamnaya-related groups carried their own genetic profile, and as they mixed with existing Neolithic populations, the genetic landscape of Europe reshuffled again. The steppe migrants carried the blue-eye allele at appreciable frequencies. As their genetic contribution spread across northern and central Europe, the allele's frequency rose. The Bronze Age did not lock in the modern distribution either. The frequencies we see today, peaking at 89% in Estonia and Finland, are the product of continuing selection, drift, and migration in the Iron Age and later. Ancient DNA samples from across Europe have now identified RS129133832 carrying individuals at multiple sites. In Sweden, around 5,000 years ago, in Hungary at roughly the same period, in Germany slightly later. Frequency in these populations rarely exceeds 10 to 15%. The dramatic increase to 50, 60, 80% in northern regions came later in the last 3,000 to 4,000 years. The exact drivers of that later increase remain unresolved. What the ancient DNA does establish is that the spread was not a single wave. It was a staggered, multi-phase process, with the allele riding multiple demographic expansions and contractions over thousands of years. What ancient DNA has not yet fully resolved is the exact migration route by which the allele moved to reach such high frequencies, specifically in the north. The candidate routes are multiple. One model traces a path from the Black Sea region northwestward along the Danube corridor into Central Europe, then northward with later population movements. Another suggests a Mediterranean coastal route, reaching Iberia early, then moving north along the Atlantic fringe. A third posits a more direct northern route, with the allele present in early post-glacial populations around the Baltic. None of these models is excluded by the current data. None is confirmed. The ancient DNA coverage is still too sparse to draw a single line on a map and say, this is the path the blue eyes took. By the time the Romans encountered the Germanic tribes, the allele had done its work. Tacitus saw. Fierce blue eyes was a snapshot of a genetic process still in motion, captured by a literate civilization and frozen into text. The Romans did not understand the mechanism. They did not know about OCA2 or HERSC2 or any gene at all. They saw a pattern in human faces and assigned it meaning Germanic, northern, barbarian, other, cultural weight placed on that neutral regulatory mutation grew heavier over the centuries that followed. In the medieval imagination, blue eyes became bound up with Northern European identity, with the iconography of saints and warriors, the literary conventions of chivalric romance. In the early modern period, as Europeans encountered the wider world through colonialism, eye colour became one of the physical markers used to sort human beings into categories. Categories that hardened the 18th and 19th centuries into racial typologies whose consequences we still live with. Single nucleotide substitution, ARS 129133832, was recruited into arguments about superiority, purity, and essential difference, all of it built on a copying error in one person 10 millennia ago that simply reduced the amount of melanin in the front layer of an iris. There is no irony quite like the sight of a 1,000-year-old cultural mythology draped over a 10,000-year-old molecular accident. The mythology feels ancient because 1,000 years is a long time for a human memory. The mutation is 10 times older, a thing that has been made to carry so much meaning. Racial meaning, tribal meaning, political meaning, is younger than agriculture, younger than the first cities, younger than the dog. Which brings us to the most unsettling implication of the whole story. Blue eyes can be this young and this accidental, and still become a marker around which entire peoples were defined. What does that tell us about the age of human physical difference itself? Blue eyes were not the only trait that arrived late. The skin tones we now associate with Northern Europe followed a separate genetic path, driven by a different set of changes in different genes, under different pressures. The single largest effect comes from a variant in the gene SLC24A5, specifically the derived allele at RS1426654. In modern Europeans, this allele is nearly fixed, present in over 99% of individuals, and it alone accounts for a large fraction of the pigmentation difference between African and European skin. Yet in every Mesolithic hunter-gatherer genome sequenced to date, that allele is completely absent. The individual from Labrania does not carry it. The 11,000-year-old Cheddar man from Britain does not carry it. Neither do the hunter-gatherers from Central and Southern Europe, whose genomes have been extracted from teeth and temporal bones. The allele first appears in Europe, in the genomes of Anatolian farmers who began moving into the continent from the southeast roughly 8,500 years ago, bringing agriculture with them. A second wave of lightening came later, carried by steppe pastoralists during the Bronze Age, who spread variants in the genes SLC45A2 and TYR. By 4,000 years ago, the modern northern European skin tone had become common, but it was not yet universal. The pale skin that seems so permanent, a feature of the north, is a composite that assembled itself in the space of roughly 5,000 years. Blonde hair tells a parallel story, with a different molecular script. Key variant lies in a regulatory region of the gene, KITLG, designated RS2821256, which dials down melanin production in the hair follicle in a manner analogous to the OCA2 switch in the iris. This allele is not a European original. It appears first in ancient genomes from Siberia, carried by population geneticists called the Ancient North Eurasians, group whose ancestry later poured into Europe through the same steppe migrations that reshaped the continent's language and its skin. By the Iron Age, fair hair had settled into the northern latitudes, but its journey to get there ran through the same corridors of migration that distributed the other traits. None of these features, the blue eyes, the light skin, the blonde hair, were bundled together at the point of origin. They were separate accidents, moving independently down separate rivers of descent, and they collided only recently, within the last few thousand years, in the bodies of people living between the Baltic and the Atlantic. The mutation for blue eyes has existed for at most 10,000 years. Homo sapiens has walked the earth for roughly 300,000, Blue eyes, therefore, occupy no more than 3 to 5% of our species' history. Light skin, in the form we see across northern Europe today, is even younger. The derived SLC2T4A5 alleles swept through European populations between 8,000 and 4,000 years ago, and the SLC45A2 variant reached high frequency later still. For the other 95% of human time, roughly 285,000 years, every face on every continent carried the darker pigmentation of our African origins. The world that built language, carved the first figurines, that painted the walls of caves, and buried its dead with ochre and flowers, was without any variation in eye colour at all. When the first blue-eyed child opened her eyes, the painted bulls of Lascaux had been on the limestone for 7,000 years. The stone circles of Göbekli Tepe, still standing today, were already ancient when her community gathered around a fire. This timing does something to the way we categorize one another, physical markers we use to sort human beings into races. Skin color, eye color, hair form are evolutionary snapshots from the very recent past. They are not the deep, fixed, ancient divisions between populations that separated long ago. They are the visible edges of processes that started, well, after Homo sapiens had already colonized every continent except Antarctica. Child born in Mesopotamia 40,000 years ago and a child born in France 10,000 years ago looked more alike, in terms of pigmentation, than a modern Norwegian and a modern Senegalese do. The difference that feels so foundational, the visual shorthand that has been used to justify segregation, slavery and genocide, is younger than many of the archaeological sites we treat as relics of deep time. The genetic architecture of race is a late draft, not the original manuscript. The palette of human appearance is strikingly plastic. Within a few thousand years, eye colour, skin tone, and hair colour can shift dramatically across a population. These traits are not bedrock characteristics. They are surface layers, shaped by natural selection, genetic drift, and the accidents of mating, always in motion. The instructions for melanin production sit in the genome in many forms, and a single nucleotide substitution in a regulatory region. One letter swapped in a stretch of DNA that does not code for a protein, but controls how strongly a gene is expressed. A crest can flip a switch that changes what a face looks like to everyone who sees it. The OCA2 variant that creates blue eyes is precisely such a switch, turning down melanin in the iris. The SLC24A5 variant that lightens skin does the same in melanocytes. The KIT-LG variant that lightens hair provides yet another independent demonstration of the principle. In each case, the genome did not need to invent a new colour. It merely altered the volume of the brown that was already there, and the structural colours, the scattering blues, the underlying pale tones, surfaced as the covering lifted. The biological pressure behind skin lightening adds a layer of physiological necessity. At latitudes above 50 degrees north, the amount of ultraviolet B radiation reaching the skin is insufficient for vitamin D synthesis in heavily pigmented individuals during the winter months, and marginals even in spring and autumn. A person with dark skin living in prehistoric Scandinavia would have needed a diet extraordinarily rich in vitamin D to avoid rickets, pelvic deformities, and the cascade of reproductive fitness costs that poor vitamin D status imposes. The shift to agriculture in the Neolithic reduced the dietary intake of vitamin D for many populations, increasing the selective pressure on any allele that lightened the skin enough to allow more UVB to penetrate and drive synthesis. SLC24A5 variant was not the sole solution, but it was the one that came to dominate, likely because it offered a large effect in a single genetic step. Other variants of SLC45A2 and TYR added further incremental lightening, but the core switch had already been thrown. Lebrania sits squarely inside the paradox. His genome, dark skin, blue eyes, dark hair, is not a contradiction. It is a snapshot of a moment before the traits we now package together had finished coming together. Vost, the light skin that marks Northern Europe today, evolved through different genes, under different selective pressures, thousands of years after the OCA2 mutation had already begun spreading through hunter-gatherer populations. The two stories were independent, tangled together only later, as the descendants of Anatolian farmers and steppe herders and indigenous foragers met, mixed, recombined their alleles over dozens of generations. By the Bronze Age, could find individuals in Central Europe carrying the blue eye allele alongside newly arrived light skin variants, but the full three-part phenotype took longer still to coalesce at the frequencies we recognize today. The fact that the La-Brania combination surprises us is not a reflection of biology. Pass. It is a reflection of how deeply modern cultural expectations have been trained on a very narrow slice of human variation, particular outcome of a process that could have resolved differently. If a single regulatory mutation can spread to 8 or 10% of the global population in 10,000 years, similar processes are ongoing right now. Human appearance is not a finished state. It is not a relic of ancient separation. It is a live, ongoing negotiation between genes, environments, and the choices people make about who to live with and who to have children with. Faces we see in the mirror are younger than they look. Every so-called traditional feature of the North, the pale blue eyes of a Baltic fisherman, fair hair of a Swedish child, the freckled skin of a redhead in Ireland, is post-Ice Age, assembled in the brief window since the glaciers retreated and still in motion. No gene fixes a face forever. The human surface is not a fixed inheritance from a distant past. It is a recent and reversible and deeply contingent work in progress. The unsettling implication is not that our physical differences are meaningless. It is that they are new. They are accidents that lived post. They are the visible residue of a few thousand years of history, not the bedrock signature of separate ancestral lines stretching back to an imagined deep time. And if these particular accidents had not survived, if the child by the fire had died without issue, if the farmers carrying SLC24A5 had taken a different migration route, if the steppe pastoralists had never moved west, the human family would look different today, and the categories we use to separate one another would have been drawn along different lines, or perhaps never drawn at all. 600 million people, possibly 800 million. The uncertainty in the count is itself a reminder that no census taker has ever gone door to door across the world asking the colour of a person's eyes. Every single one of those individuals carries 2 copies of the same single nucleotide, polymorphism RS12913832, at precisely the same position in the regulatory region of the HERC2 gene on chromosome 15. The mutation is identical in all of them. Not a family of similar mutations, not a recurring pattern that evolution happened to favour in different places. One molecular event, in one cell, in one developing egg or sperm, one person, roughly 10,000 years ago. The geographic span of that accident is now planetary. A fisherman on the Lofoten Islands pulling cod from the North Sea in the grey winter light carries it. A child in the mountains of Afghanistan walking a dirt path to a school built of stone carries it. A classroom in Dublin in late autumn. 30 children bent over copybooks, half of them with pale eyes, the colour of a cold sky. The same switch, flipped once, echoes in every iris. Village in Turkmenistan, where blue eyes are rarer enough to be remarked upon, but present nonetheless. The mutation does not respect the borders we have drawn. It cuts across languages, nations, religions. The stories tell themselves about who they are and where they come from. Consider what this specific genetic fact does to the idea of ancient biological boundaries between populations, trait that has been held up by Roman ethnographers, by medieval chroniclers, by 19th century race theorists, by modern cosmetic advertising. As a marker of a particular people, a sign of Northern or Western European identity, originated in a single individual, host. It was not the accumulated signature of a tribe that lived in isolation for tens of millennia. It was not the evolutionary inheritance of a distinct branch of the human family that diverged from other branches in deep prehistory, was one person. And it spread through migration, and chance, and mate choice, and the silent passage of a recessive allele through countless generations of brown-eye carriers who never knew what they were carrying. The notion of blue eyes as the ancient patrimony of any specific population collapses under the weight of the molecular data. What looks like a deep ancestral divide is, on inspection, a very shallow and very specific accident that happened to survive. The recessive inheritance of RS 12913832 adds another layer to the scale of its hidden reach. For every blue-eyed person walking the earth, every homozygous carrier of the derived allele, 2 copies of the switch in the off position, there are many more people carrying 1 copy. Heterozygotes, brown-eyed individuals who have no outward sign of the mutation, but who carry it in their genomes, nonetheless, a silent passenger on chromosome 15. They will pass it to roughly half their children, and if they have children with another silent carrier, some of those children will open their eyes and reveal a colour that neither parent displayed. The mutation is more widely distributed than blue eyes themselves reveal. It flows beneath the surface of the visible world, threading through populations that consider themselves entirely brown-eyed, waiting for the right combination of parents to surface again. Trait you think of as rare, as distinctive, as belonging to a specific kind of face, is in fact present, quietly, invisibly, in millions of people who would never be counted in the statistics. If a single neutral mutation, a loss of function, tweak in a regulatory element, near a pigmentation gene, can spread to define the appearance of an entire continent in 10,000 years. If it can slip across mountains and rivers and language barriers, carried silently in the genomes of people who had no idea it existed, then how many other silent mutations are moving through human populations right now. Mutations that do not touch pigmentation but shape something else. The wiring of a neural circuit, the strength of a bone, the way the immune system reads a particular pathogen, the age at which a girl first menstruates, the subtle architecture of a personality, traits we take to be permanent and defining. And ancient may, in fact, be younger than the invention of writing. We have sequenced the genomes of living people in enormous numbers, but we have only just begun to sequence the genomes of the dead, and it is in the bones of the dead that the timing of these changes is recorded. What this story does not resolve, what genetics cannot resolve, is the question of what a trait means for human identity. Fact that blue eyes are genetic does not make them essential. The fact that they are inherited does not make them ancient. Fact that they are shared by 500 million people does not make them a foundation for any claim about who belongs, where, or whose ancestors were, native to which soil. A genetic trait can be recent, accidental, and contingent, and blue eyes are all three. The molecular evidence is unambiguous on this point, even if the cultural habits that load eye colour with meaning are slower to shift than the science. The image to hold as the story settles into its final shape is the child by the fire. She is perhaps 4 years old, dark-skinned and dark-haired, sitting at the edge of a circle of adults whose faces are lit from below by the flames. Her own face is turned slightly away from the heat, and the firelight catches the pale blue of her irises, colour that no one in her community has ever seen on a human being. She does not know she is different. She does not know that 10,000 years later, 500 million people will carry the accident written into the cells behind those eyes. She is simply a child, watching the flames, waiting for sleep. If a tiny switch in a regulatory gene can connect 500 million strangers across 10 millennia, what else do we all share that we have not yet found? A story that began with a single child by a fire 10,000 years ago ends, for now, not with an answer, but with a question. The genome is still being read. Bones that could answer. The open-ended questions about location and mechanism may still lie under meters of sediment in a cave in the Caucasus, or under a shell midden on a Black Sea coast that no longer exists, or under a field in the Balkans that has never been excavated. Science is mid-sentence here. The story has no final punctuation. The image to hold, as the screen fades, is the child by the fire, 4 years old, dark-skinned and dark-haired, her blue eyes reflecting the flames, entirely unaware that she is the beginning of a question that 10,000 years human history have not yet answered. We know she existed. We will never know her name.